seminal vesicles, the prostate gland, and the vulvourethral glands. They all contribute to seminal fluid that mixes with the spermatozoa to comprise the semen. Now, it turns out that, by the way, the spermatozoa are sensitive to acid. And so you find that the seminal fluid is alkaline. So the seminal vesicles secrete an alkaline fluid that accounts for 60% of the volume of the semen. So they make the largest contribution of the three glands. Okay, so what's in it? One, fructose. Fructose is the main fuel source used to generate energy, ATP. Spermatozoa need energy to swim, so that's not a surprise. Then, the seminal vesicles contain a coagulating enzyme that keeps all of the spermatozoa together, so that during ejaculation, everybody passes from point A to point B together into the vaginal tract. It's kind of like when you're in kindergarten and you're crossing the street, everybody holds hands, cross the street together. Okay, you all get there safely. Coagulating enzyme. Okay. And then finally, it contains prostaglandins. These are chemical mediators that decrease the viscosity of the cervical mucus. The cervical mucus sits within the cervical canal. This is the uterus. It's usually a plug of cervical mucus that sits right here that serves to block the entrance into the uterine cavity. This mucus tends to be thick, okay? And it tends to, in a sense, shut the door. Well, the prostaglandins thin the viscosity and make it easier for the spermatozoa to pass through. The prostaglandins also stimulate reverse peristalsis. In other words, when the uterine contracts, think of childbirth. It contracts from fundus down the cervix like this, okay, so that any contents are expelled. Well, with reverse peristalsis, it assists with bringing the spermatozoa into the uterine cavity. Just the opposite, the direction of the contraction. Okay. Prostate gland makes up the second largest contribution, about 20 to 30% of the volume. It also contains a nutrient, the citric acid, but it's not the main nutrient. There are several enzymes present within the prostatic fluid, including fibrinolysin. So at some point when the spermatozoa reach the female reproductive tract, fibrinolysin breaks down those bonds between the spermatozoa and let them go their individual ways. What you'll also find within the prostatic fluid is acid phosphatase. The only reason I bring this up is that sometimes acid phosphatase is used as a marker for metastatic prostate cancer, particularly metastatic cancer to bone. Okay, it's a reflection that metabolically active prostate cells produce this particular compound. It also contains prostate-specific antigen. Well, PSA is used as a screening test for prostate cancer. Okay, again, this is a normal substance produced by prostatic cells. And finally, the third accessory gland is the vulvourethral glands that sit within a urogenital diaphragm. These actually produce secretions that are released in advance of orgasm. Okay, in fact, they're released during what people would call the arousal phase of the sexual encounter. And this is an alkaline substance that's released into the male urethra that serves to neutralize any acid left behind by the urine. So that it kind of paves the way for the emission of spermatozoa. We said that the spermatozoa would be harmed by the acid environment. So this alkaline fluid kind of protects the spermatozoa by neutralizing that acid before ejaculation. Now, let's suppose you have a couple. Let's suppose you have a couple who wants to achieve fertility, and they're having difficulty, and they decide that they're going to go to a fertility clinic. What do you think that, in about 50% of cases, by the way, the cause is the female, and about other 50%, the cause is the male. Who do you think it is that they test first? Why would that be?
Because of the sperm count.
Well, the sperm count is a cause of infertility, but why would they test the male first, as opposed to testing the female?
Maybe because the semen's not getting to.
Wait, you're telling me mechanisms for infertility, but why would they test the male first? Because you could say, well, maybe the fallopian tube's blocked and the egg's not reaching.
Is it easier to test the male?
It's not only easier, but it's what?
Fast.
It's faster, and it's less expensive. So, you may be able to test the male. It might take, 15 to 20 minutes, whereas the female component of the evaluation can go on for months. Okay? Whereas the evaluation of the male may take $150, $200. The female workup may go into the tens of thousands of dollars. So if you could rule out the male first, that would be the thing to do. You may save some money. So what they're going to do for the male is do a semen analysis. They're going to obtain a sample of semen and evaluate it. One of the things they evaluate is volume. Okay. In terms of, let's say of spoons, teaspoons, tablespoons, cups. What's a normal volume for semen from an ejaculate? Yeah, but that doesn't answer the question. In terms of teaspoons, tablespoons, cups, how much is that? What would you think?
Not a cup.
Well, how much is, well, you see 2.5 to 5 milliliters, but how much is that? That's the question. Is that, for instance, a couple of tablespoons?
That's
One
what I was
tablespoon.
thinking. That's what I was thinking, honestly, just a couple of tablespoons. But I don't know.
Yeah,
if
Chris,
that's
come on,
right.
tell us.
I don't think it's.
Come on, you never know.
It's about a teaspoon.
Oh.
Oh, actually,
And
I thought it was more than that.
that's you.
so sometimes the problem is simply inadequate volume. The analysis will include a sperm count, normal being between 50 to 150 million per milliliter. So if you produce 5 milliliters, each milliliter has about up to 150 million spermatozoa. Okay? Whereas less than 20 million per milliliter is associated with infertility. The reason is that although you have millions and millions of spermatozoa that are released into the vagina, you may have only 50 to 100 make it into the fallopian tube. It's a hostile environment out there. The tendency is during female orgasm for the uterus to contract, and people have described it like the spin cycle of a washing machine, where any spermatozoa making it up into there oftentimes get ejected. They have to pass through the cervical mucus, they have to pass through the body of the uterus, and then they have to make it up into the fallopian tube. So only about 50 to 100, so you need that type of ratio, and it's a tremendous ratio, in order to achieve adequate numbers to achieve fertilization. And then the other important thing is that the semen has to be alkaline. Okay? The spermatozoa don't develop well in an acid environment. What do you think they can advise a couple if the volume of the ejaculate is too low? Do what?
Something to increase the sperm and the egg. Drink more water.
Drink more water. No, that's not it.
You know what I said.
It's not sperm count, we're talking about volume here. Well, what they're telling them to do is abstain. In other words, what you want to do is time fertilization. By the time and then for 2 weeks before that period of time, that window, you want to abstain from having sex. And that will build up volume. Whereas if, married couples, newly married couples, how many times a day do they have sex? Or how many times a week, let's say? At least once a week, you think?
No, at least once a day. That's what, but you know.
About 15 or 20 years ago, there were studies that showed that many married couples had sex 2 to 3 times a day. So you can imagine in terms of volume that would impact volume. Now, if you've been married for a longer period of time, of course, those numbers go down. What about sperm count? What kind of questions would you ask if someone had a low sperm count? How about boxers or briefs? Why would that impact anything? Because of temperature. So if you wear briefs, okay, you bring the testes up closer to the core body temperature, and that can impair development of spermatozoa. What do you do for it if the pH is too acidic? What can you do there? Change diet. Now, what they'll do when they do a semen analysis, they'll look at other things too. They'll look at morphology. For instance, you'll see some spermatozoa that have 2 heads or 2 tails. Well, it turns out that if you have a high percentage of those abnormal spermatozoa, that your risk of infertility is higher because those abnormal spermatozoa don't tend to fertilize. You look at motility. When you look under a microscope at a fresh specimen, there are always going to be a certain percentage of those spermatozoa that are not swimming. Okay. Of course, if they don't swim, they're not going to fertilize. So there are a lot of things that, in fact, they look at. But the evaluation of the male really doesn't take that much time, and it also doesn't cost very much money. So they would want to rule the male out first before embarking on an evaluation of the female. What do you think is one of the most common causes of infertility in women?
Not being attached, like the sperm not being attached to the
PID, pelvic inflammatory disease. Someone's had a sexually transmitted disease like chlamydia or gonorrhea, and it can lead to scarring of the fallopian tubes. And so even though they're producing eggs, those eggs have a hard time passing through the fallopian tube, or for the spermatozoa and the egg to meet up. Okay. All right, what we see here is, this is an electron micrograph at the top of a cross-section through a seminiferous tubule. Here's one seminiferous tubule, here's another, here's another. And what we see here is that the spermatogenic cells line the wall of the seminiferous tubule. This boundary right here is the basement membrane. Okay. So sitting against the basement membrane are cells called spermatogonia. Single would be spermatogonium. A spermatogonium is a stem cell. Stem cell. Does anyone know what stem cells are? What is that? Yeah, they're non-specialized. They're undifferentiated cells. So these stem cells, these spermatogonia, have a normal complement of chromosomes. But one thing about stem cells is they undergo fairly frequent mitosis. And when they divide, they produce 2 types of daughter cells. Oftentimes these are referred to as type A and type B. The type A daughter cell By the way, both of these will have 46 chromosomes because this is mitosis. The type A daughter cell simply replaces the parent and continues to undergo fairly rapid mitosis. Whereas the type B daughter cell will then differentiate and divide further, and it can differentiate along one or multiple lines. Okay? So it's going to develop, it's going to become more mature. Depending upon the type of stem cell, it may only differentiate along one line. In fact, in this case, it would become a spermatozoa. If it were a stem cell within the bone marrow, it could differentiate along multiple lines. It would be more, we call it multipotent. You know, stem cells are classified as either unipotent, multipotent, pluripotent, or totipotent. And that classification is based upon how many different lines it can differentiate along. So for instance, in the bone marrow, those stem cells can become either red blood cells, platelets, or any of the different types of white blood cells. So those are more pluripotent. Whereas here, these are only going to differentiate and become spermatozoa. So sitting along the basement membrane, you have your spermatogonia. Okay. If it were to divide, let's see what it shows here. Okay, it shows the type A daughter cell here, which is going to hug the basement membrane and simply replace the parent. And here would be the type B cell. The B cell is going to differentiate into what's called a primary spermatocyte. A primary spermatocyte still has 46 chromosomes. But it's the first of the spermatogenic cells to undergo a specialized type of cell division called meiosis. Meiosis is a specialized type of cell division where the daughter cells have half the number of chromosomes as did the parent cell. So with mitosis, the daughter cells have an equal number of chromosomes. With meiosis, the daughter cells have half the complement, have half the number of chromosomes as the parent. How many chromosomes do our cells possess?